We're going to start the second half of the workshop, and one brief announcement. You'll notice that while you were out for the break, some evaluation forms appeared in front of your chairs, and we would very much appreciate it if you would complete the evaluation forms and leave them on the table out in the lobby. It's very important for us to get feedback on these workshops. So I'm going to introduce the next speaker, who is Dr. Leo Gordon. He's a hematologist-oncologist, and his specialty is the treatment of lymphoma. And, here's maybe Dr. Lick will do the introductions.
Hey, John.
Thank you. We're going to start again. We're very pleased to have here today Dr. Leo Gordon, who's the Friend Professor of Cancer Research in the Robert H. Lurie Cancer Center and Division of Hematology Oncology. Dr. Gordon is an internationally known expert in the treatment of the tumor of white cells of specific antibody and other types of white cells. Together, collectively, these are tumors called lymphomas. Dr. Gordon will be talking about some of the biological approaches to better understanding these types of tumors and how a lot of treatment progress has been made over the coming years. Do you have your microphone?
I'm good. I think so. Can everybody hear me okay? Yeah. Great. Thank you. Thanks, John. So I'm going to spend the next 25 minutes or so talking about mostly biologic therapy. It's titled Biologic and Cellular Therapy, but I know that Tim Kuzel talked to you a little bit about cellular therapy as part of understanding immunotherapy. So this is what we're going to talk about. We're going to try and get through all of these sort of topics kind of as an overview. And we'll start a little bit about talking about some of the techniques that are used to understand pathways. We'll talk a lot about pathways, cellular pathways that serve as targets for therapy. They also serve to give us a better understanding of a number of diseases, not just lymphomas, but a lot of other cancers as well. But we have to know a little bit about what I call the molecular determinants of outcome, how we use molecular genomic information to predict outcome. We'll talk about innate tumor immunology, innate immune responses, that is the T-cell responses. We'll talk about monoclonal antibodies, which are now being used to treat a variety of malignancies, certainly lymphomas. We'll talk about vaccine therapy, where it is today. Is it still have a role to play? Then we'll kind of focus on a number of pathways. Cell death pathways, we'll talk a little bit about apoptosis, or programmed cell death. And then we'll talk about specific pathways, NF-kappa B, an important transcription factor in cell growth and cell development. We'll talk about the IMiDs, thalidomide and Revlimid, lenalidomide as potential treatments for a variety of disorders, histone deacetylases as targets, and then an interest of mine, the whole role of hypoxia and how it might influence cancer progression. So first of all, a lot of what we're doing is based upon observations that have been known for the past maybe 15 years or so, and that is the idea, and you may have some of this information already, but the idea that you can do what are called gene arrays, where you take known genes, DNA, and put them on a chip and try to hybridize them with patient samples, RNA from patient samples. So you have, for example, lymphoma biopsies going in this direction, you have known genes going in this direction, and you can label those when there's hybridization into what's called a heat map. This is a heat map where the red color represents binding or a lot of activity of that gene in that particular set of biopsies, and the green represents low activity of genes, representing the absence of genes in a particular set of malignancies. This is used a lot in lymphoma, but it's used in breast cancer, it's used in a variety in lung cancer, sarcomas, it's used in a variety of other malignancies to sort of get a sweep of what the genome looks like in a particular set of tumors. And what you start off with is something that looks like this, but by dissecting it and using bioinformatics, you can actually get a lot of information, much like you can when you sort of dissect a painting that's perhaps got some a drawing on it that obscures the real painting. So ultimately, you get information that makes sense. And this is sort of the analogy that I think we can use. How does this used clinically? There was a paper published in maybe about now 12 years ago looking specifically at patients with large cell lymphoma. That's an aggressive type of malignant lymphoma. And people did this genome analysis and found an important observation, I think, and that was that one set of patients, as described as the germinal center-like lymphomas, that's because they had genes that were present in germinal center development, germinal center of the lymph node, and another set of patients that had what's called an activated B-cell-like gene signature. These are basically gene signatures. And interestingly, the patients that had the germinal center signature did a lot better, this is a survival curve, than the patients who had the activated B-cell-like signature. So now we've taken a step where we used to say, well, if somebody comes in with large cell lymphoma, we look under the microscope. Most of them look very similar. They don't look a whole lot different. You look at the patient. Patient may have some symptoms, may not have symptoms, and we think we can predict, based on how people look and what the biopsy looks like, how somebody's going to do. And we can to a certain extent. But this takes us, I think, one step further to the molecular level, where we can begin to predict outcome based on the gene signature. And not only can we use this as a predictor of outcome, but I think also we can begin to identify genes in these signatures that we can begin to target for potential therapy. So, for example, this is follicular lymphoma. This is another type of lymphoma that's considered to be a low-grade lymphoma, not as aggressive as the large cell lymphoma. And we can identify under the microscope, this is biopsy specimens, different grades of the tumor. As you might expect, the higher the grade, the worse the tumor ought to be. So you go from grade 1 to grade 2 to grade 3A and 3B. It stops at 3B. Why 3A and 3B? Because pathologists have a hard time distinguishing these, actually. And we would think that the higher the grade, the more aggressive the tumor will be. So people applied. Actually, the person that published this was a former graduate of the college here and the medical school here, Sandeep Dave, who published data that suggested that if you do this gene signature on these follicular lymphoma patients, you wind up with a group of genes called immune response genes that represent T cells, which help to regulate the immune response, and you find that those patients had a favorable prognosis based just on this gene array. And if you look at another group of patients, you have another group of patients that have genes that are called immune response 2, and these are mostly macrophage genes. So what you wind up with are two patients that look like this. Now we're looking at the patient not from what they look like on the biopsy or what they look like on the physical exam in the room, in the bed, but rather what they look like when you do gene analysis. And what you have are follicular lymphoma shown in blue in patient 1 on the left. And in this patient, you have very few macrophages, and you have in pink a lot of T cells. This patient turns out to have a longer survival than the patient on the right, patient number 2, who has the same number of follicular lymphoma cells in blue, but a lot less T cells in pink and a lot more macrophages, a patient now with a shorter survival. So we can begin to predict, at least in this set, that the T cells, these T cells, which help to regulate the immune response, might be important in outcome. So what's the consequence of that? Well, one consequence is to develop a treatment that may increase the T cell numbers in patients. So how do we do that? We know, for example, that T cells have built into them sort of a programmed cell death mechanism where they die off at a certain rate. And that's regulated by a gene and a protein on the surface of the cell called PD1, programmed death 1. So people have now developed, and we've worked with, an antibody specifically designed against PD1 to block the activity of PD1 and increase the number of T cells and also natural killer cells that are present in the blood. And so we applied this idea to a group of patients with lymphoma who had a bone marrow transplant, and we gave this antibody to those patients, 70 patients from around the world, and we found that the outcome of that group of patients, all we can say about it is that it was better than we might have expected. We really have to do you heard a little bit about probably randomized phase 3 trials earlier. You have to do a randomized phase 3 trial to know whether it really made a difference. But we also had some information that really suggested that it might make a difference, and that is the following, that if we measure T cells in the blood immediately following the administration of this antibody, we find a very striking increase within 24 hours, and that increases over time, an increase in circulating CD8 and central memory cells, and those are cells that help to control the immune response and help to fight off malignancies. So by just giving this antibody, we're able to increase the number of the central CD8, what are called CD8 positive memory cells, and that may be an explanation for why patients do better following this antibody. So obviously a lot more work needs to be done with that. So I mentioned antibodies. The antibody we used here is called CT011, and this is a chimeric, what's called a chimeric antibody, which means that it's an antibody somewhere in between a murine or mouse antibody and a human antibody. And what's the difference? What are some of the differences? And you may have heard a little bit about this, but what we can see from this slide is that there in fact are some differences. The murine antibodies are more immunogenic. They elicit more of an immune response, so the body tends to fight off these murine antibodies. Also, they are cleared, probably because they're more immunogenic, they're cleared more rapidly from the blood. Those are things we don't want so much. We want perhaps a little bit less clearance, so the antibodies are hanging around longer. And so we now make antibodies that are half mouse, half human, and ultimately totally humanized antibodies. And many of the antibodies that we're using today are either chimeric or full humanized antibodies. And you'd expect those to be less immunogenic, have less clearance, and have more toxicity against the tumor cell. And so that's what this is saying, that they have a longer circulating half-life, they're less immunogenic, but also importantly, they have more potent ADCC. So what's ADCC? Is antibody-dependent cellular cytotoxicity. So what we think, the way we think some of these antibodies work, and I'll show you a drawing of this in a second, is that they don't necessarily kill the tumor cells themselves, but they recruit T cells, we heard about T cells, and natural killer cells to do the job. And that's what ADCC is, and then CDC is complement-dependent cytotoxicity, and I'm not going to talk too much about that. We'll talk about ADCC. So let's hear about some of the antibodies that are actually used clinically today that function in this way. So one of them is a drug called Rituximab, which is an antibody that targets a protein on the surface, and the protein is shown in these spikes here, on the surface of almost every B-cell lymphoma. So there are 70,000 new lymphomas in the United States a year. 90% of them are B-cell lymphomas, maybe 5 to 8% are T-cell lymphomas, and the remainder we don't know, we can't tell. So we're talking about B-cell lymphomas, and the B-cell lymphomas are characterized by this CD20. CD stands for cluster designation. It's just the nomenclature for the many proteins that are present on the surface of malignant cells. Actually, this CD20 is also present on normal cells, but people thought, well, if we have an antibody against this CD20, maybe we can do some damage to the tumor cell without, hopefully, much damage to the normal cell. So why CD20? Well, first of all, it's expressed in most B-cell lymphomas, and interestingly, it doesn't do what many of these targets do. Once they're exposed to an antibody, they shed or they internalize, kind of like a turtle or they modulate their expression. CD20 just stays there and says, hit me, basically, is what it's saying. So, which is nice, because that's a nice target. And so here's the antibody. Rituximab was developed in a number of laboratories, at Dana-Farber and at Stanford. And we did some of the early clinical trials with this antibody in the mid-1990s. This is what a typical antibody is going to look like. It's got its functioning part here, which is, in this particular antibody, is murine, and it's got what's called an FC domain, which is the human part of the antibody. This part sticks to the CD20, and this part does something else. What does it do? It causes, it allows ADCC to go on, because if you, I don't know if you can see this very well, but if you look right in here, you see this part of the antibody in yellow sticking to the red target on the tumor cell, but it's the other part, its other end, the FC domain, is binding to either natural killer cells, T cells, monocytes, all what we can sort of term effector cells. And so this is what ADCC is. This antibody is recruiting this cell to kill that cell. So that makes sense. Why does it work? It works because you can see this natural killer cell, or this T cell, can directly kill tumor cells, and there's a lot of research about how it works. But why doesn't it work all the time? I mean, that's a question that I think is worth asking. It should work 100% of the time, right? So it doesn't for a variety of reasons. There's some theory that there are mutations that occur right here, right where this FC binds to this receptor on the cell. And people are now beginning to study these mutations for explanations about why Rituximab doesn't work all the time. So this is an example of how understanding the basic biology can kill tumor cells. Before I came in, I was talking to somebody that was interested in radiology, and you heard a little bit about radiology. You heard about microspheres that are labeled with yttrium to kill hepatocarcinoma cells, right? So this is the same idea. If you think about it, this is taking that Rituximab antibody that I just showed you and saying maybe we can arm this, make it a better antibody by attaching a radioisotope. And we've used both the same yttrium-90 that you heard about on the microspheres and also I-131, radioactive iodine. And those attach to this part of the molecule, or here on this part of the molecule. Both of these drugs are available clinically. And this is the one, this drug called Zevalin is the drug that we've used quite a bit, and we helped to work on this drug to develop it to get FDA approval. And the way it works is, I think can be shown in this slide. If you look at two lymphoma specimens, for example, if you look on the left, you get naked antibody. If you can see the yellow. But it binds mostly to the, this is going to just run its course here. I'll let it finish, then I'll talk about it. If you look on the left, it's binding to the tumor cells that are on the outside of this tumor. You can't get to the stuff in the middle because the tumor is too big. But if you use radioimmunotherapy, what you have is you have binding to the outside cells, and then you have the radiation shooting across and getting the remainder of the cell. So there's clear data now that show that using radioimmunotherapy is better, probably, in terms of tumor response than just using the plain Rituximab. Problem, of course, there's always another side to it. The problem is probably it's got more toxicity. So you have to kind of always balance the benefit with the potential toxicity of any new therapy that we're working with. So this is the concept for radioimmunotherapy. So I said I would talk a little bit about vaccines. A hot area in cancer research, and specifically in lymphoma research, work of a number of investigators. Ron Levy at Stanford did a lot of the work on this. And basically the idea was that if you can make vaccines against infectious diseases, polio, and a variety of other disorders, you can also maybe make a vaccine against cancer, against the tumor. Well, so the idea was to take a biopsy from a patient, work in the laboratory to make a vaccine with a variety of different approaches, and I won't go through those. But you make the vaccine, and the vaccine is actually made to something that's very unique to the tumor. There's a protein called an id protein, an idiotype protein, on the surface of a lymphoma cell that's very unique to that individual's lymphoma. So this is this is a vaccine that's made specifically against one individual. It's not a generic vaccine like the polio vaccine might be. It's made specifically against the tumor. So you have to have the biopsy specimen, and to make the vaccine, you then have to throw in something that increases the immune response, some kind of what's called an adjuvant, and then you give it back to the patient. Makes a lot of sense, and the early trials suggested that there was a lot of activity, but when it was subjected to the real test in clinical cancer research, and in any medical cancer research, which is a randomized phase 3 trial comparing this to some kind of control, not quite a placebo, but a non-specific vaccine, it turns out that there wasn't a whole lot of difference. So what's the reason for that? First of all, what antigen are we targeting? We may not be targeting the right antigen. How do we deliver the vaccine? Is just injecting it intravenously the right way to do it, or are there better ways to do it? There probably are better ways to do it. Are we using the right adjuvant or immunostimulant to stimulate the immune response? We may not be. And how to identify good response? Ultimately, we want to see people cured of their lymphoma, and that's really the true test of this. But so far, that hasn't happened. I think there still needs to be a lot of research that goes on that tells us how to do this better. So I'll show you some of that research in the next several slides. This takes advantage of the fact that tumor cells and all of our cells in the body have a natural built-in, what we call programmed cell death, meaning they die at a certain point in time. And one of the problems with certain types of cancer, especially some lymphomas, is that they don't die soon enough because the mechanism for cell death has been interrupted in some way. So this is a pathway, very common pathway of cell death, and we won't go through it in detail. It involves the mitochondria in the cell, and it involves a bunch of proteins called caspases, and ultimately what's been referred to kind of as the executioner caspase, caspase 3, which eventually leads to cell death. And when you see a pathway, whenever you see a pathway in an oncology lecture, when people are thinking about our targets along that pathway, we want to see if we can interrupt or increase the pathway somewhere along here. And all these drugs that we're showing you here are things that do that. They interrupt the pathway in one way or another. On the left is a cell, a normal cell, that's undergone apoptosis, and on the right is a cell, a tumor cell, that looks intact, that hasn't undergone apoptosis. It's surviving or evading apoptosis, and we're trying to figure out ways to keep it from doing that. Another important pathway is something called the NF-kappa B pathway. NF-kappa B is a transcription factor that helps cells grow and divide, and it's something that in tumor cells we actually would like to maybe get rid of or interrupt in some way. And the main point on this slide is that drugs that interfere with this NF-kappa B, and there's a drug called bortezomib, which has a lot of activity to block NF-kappa B, is active in myeloma, and there's some in multiple myeloma, and there's a lot of evidence that it may also be active in certain lymphomas, both B-cell lymphomas and the rarer T-cell type lymphomas. So that's the major point of the slide. Well, let's show you how, this is one mock-up of how this might actually work. This is a cell, and this is the outside, this is the membrane, and this is the inside of the cell. And we know that there are a variety of proteins which are called IKK proteins, alpha, beta, and gamma. And when the cell is stimulated by a variety of things, any kind of outside stimulus, virus, chemotherapy, radiation, a variety of stimuli, these proteins get phosphorylated, so a phosphorus group gets attached to them. And when that happens, they stick to NF-kappa B, making it inactive. So in this state, NF-kappa B doesn't work, and it can't cause the transcription of certain genes, the activity of certain genes. It can't cause cell growth. So this is sort of an anti-cancer state. But what happens is that what happens to many proteins in the cell is that they get tossed into the trash. And the trash for the cell is something called the proteasome. There's a variety of chemical reactions that take place that cause these I-kappa B proteins to get tossed into the trash and leaving NF-kappa B unprotected, active, and able to cause transcription. So the concept is, how do we prevent that from happening? We use the drug bortezomib or Velcade, blocks this proteasome and blocks destruction or tossing of this I-kappa B protein, leaves it attached to the NF-kappa B, and NF-kappa B is no longer active. So understanding that basic biology has led to the development of a drug which leads to marked improvement in the results of treatment of patients with a variety of disorders. And so that's the basic mechanism for that. There's another set of drugs which are interesting, which help modulate the immune response, and we consider these biologic drugs, and those are the drugs thalidomide and lenalidomide. And you've heard about thalidomide, if many of you haven't experienced it, but you've heard about it because in the 50s and 60s it was a great sleeping pill. Thalidomide, in fact, causes drowsiness. Unfortunately, people didn't recognize that when pregnant women took thalidomide, they were pregnant when they took it, there were some huge major birth defects that occurred. The drug was put on the shelf, and all the research that had been done with it before, recognizing that it was an immune modulator, kind of went down the drain, rightly so. But I think that once it was recognized that you could avoid those problems simply by not having pregnant women take the drug, it came back to study, and in fact a new version of the drug, lenalidomide, the 2010-11 version of thalidomide, was developed. And this drug inhibits angiogenesis, which is the production of blood vessels for tumors. It augments the T cell again, the T cell I showed you before, and in a variety of different ways. So an important drug. It's active in a variety of malignancies, myeloma, something called Waldenstrom, something called mantle cell lymphoma, malignant melanoma, a non-hematologic tumor, and certain types of leukemia or pre-leukemia. So these drugs are active. How do they work? Well, this is a myeloma cell on the top. This is what's called a stromal cell, which is sort of the backbone cell that's in blood vessels and in tissues. And there's clearly an interaction between cells that circulate in the blood and the supporting cells. And when there's that interaction, they interact by binding to receptors here. Something happens. You get the production of a variety of growth factors that stimulate the growth of the malignant cell. So the stromal cells in the body stimulate the growth of the malignant cell, and you get this sort of crosstalk that occurs right in the neighborhood of the tumor. So how does Revlimid and thalidomide work? It blocks that interaction between these two cells. So you no longer get these growth factors that cause the growth of these malignant cells. It also turns out that lenalidomide especially activates caspases. I showed you those caspases that cause apoptosis. Lenalidomide activates those, leading to more cell death. Clearly an important step. And then in the last maybe several slides, last few minutes, I'm going to talk a little bit about something called HDAC inhibitors, and then talk a little bit about oxygen and the role of oxygen. So HDAC inhibitors are really important in that they target two sets of proteins or enzymes, something called histone deacetylases and histone acetyltransferases, which are important in DNA transcription. And turns out that if in the normal state is for histone deacetylases to take acetyl groups out of the DNA strand, basically, leaving them much less likely to be transcribed. if you have an inhibitor of these histone deacetylases, you leave the acetyl groups in place, and you have a much smoother way toward transcription. So it turns out that these histone deacetylase inhibitors, or HDAC inhibitors, have a lot of activity in a variety of different tumors, including leukemias, myelomas, and lymphomas, by altering gene transcription. So I think an important advance, and they do, this is just a slide showing other things that these HDAC inhibitors do. They increase death receptors, they increase apoptosis, they decrease angiogenesis, so they decrease the flow of blood to the tumor. So clearly, a lot of clinical trials are now ongoing with these histone deacetylases. Then finally, a word or two about hypoxia. We live, as you know, in an oxygen-rich environment, so it makes a lot of sense that our response to oxygen and lack of oxygen is going to be important somehow. And you might imagine also that tumor cells' response to oxygen and lack of oxygen is important. So back in the early part of the century, Otto Warburg noticed or postulated that tumor cells are able to get around the lack of oxygen. You might imagine a big tumor mass is probably hypoxic in some way, has little oxygen, doesn't have enough oxygen. How does it survive? And there's data now that suggests that these tumor cells shift their metabolism from oxygen to glucose. So they're shifting their metabolism in an entirely different way, relying on the hexose monophosphate shunt. And how does that happen? How does it do that? There are a group of proteins, genes and proteins, that are called hypoxia-inducible factors, and there are a variety of these that do basically that. They are able to make us respond to lack of oxygen. So under normoxic conditions, when the oxygen levels are normal, these proteins are inactive. So how are they inactive? Well, that's actually known. This is the HIF. This is an example of this HIF-1 alpha. It gets hydroxylated by something called proline hydroxylase and allows it to bind to a variety of other proteins, and then, just like I showed before, tossed in the trash into the proteasome and degraded, not present, so we don't need it under normoxic conditions. Under hypoxic conditions, what happens is that this proline hydroxylase is blocked or inactivated, doesn't bind to this HIF. This HIF is no longer able to be destroyed by the proteasome, so it gets activated and is able to lead to a variety of targets which are important in cell growth and important in our response to hypoxia. And this is just an example of the different pathways that are targeted by this HIF. And it's not at all surprising that this is important, because HIF is regulated by oxygen, and if you think of oxygen as a fundamental regulator of cell development and cell growth, the fact that it controls all of these things is really not a surprise. And so these all become, so HIF now becomes a target, a biological target for cancer therapy, and that's something that we're actively involved with. So, last slide. So we've talked about molecular determinants of outcome and how these gene sequencing, gene arrays can help us predict outcome. We've talked about how they may predict T-cell responses. We've talked about monoclonal antibodies, especially the ones that may increase T-cell responses or others that may cause cell death in lymphoma cells. We've talked about the role of vaccine therapy, cell death pathways, NF-kappa B pathways, the IMiDs, now resurrected as an important agent in a variety of cancers, and then the HDACs and the role of oxygen. So I'm going to stop here, and I don't know if there's time for maybe a couple of questions. All right, thank you very much.
Thank you, Leo, for that comprehensive review. This is open for questions.
Yes.
So I was curious when you mentioned the HDACs, and I was wondering if there are other drugs that have epigenetic targets, either methylation drugs or nucleosome positioning drugs or anything of that sort.
There are, and so the whole role, and this is actually something that John has focused his research on, the whole concept of not the genetics, but what is considered to be the epigenetics. So DNA methylation, histone deacetylase. So there are a variety of drugs which are both in clinical trials and also now approved for clinical use, which are epigenetic modifiers. So yes, absolutely, and those are important drugs right now. But we're looking for better ones, but those are important drugs. Yes.
Other questions? Leo, of all these, what do you have the most I know you're very interested in, and I think it's very interesting the HIF factors. Where do you think the next breakthrough in this disease is going to come?
In lymphomas?
Yes.
So, I think that two things. One is understanding subgroups of patients that are defined by the genes that they express. And I think that the concept that you can target the genes that are expressed, I think we're going to have much more focused, and I don't necessarily use the term individualized cancer therapy. I'm not sure exactly yet what that means, but I think that targeting genes that are present in some patients with lymphoma and not present in others is going to lead to an alteration in the outcome. And I think that's where the next big advance is going to come from. Not from new chemotherapy drugs, at least in this disease, and maybe not even from new antibodies, but from small molecules that may be able to target specific genes and pathways.
Any other questions? Well, thank you very much,
Thank
Leo.
you. All right. Thanks very much.